Okay, now we're going to look at myoglobin and hemoglobin. These two proteins carry oxygen, or molecular oxygen, O2. We'll first look at myoglobin. This is a monomeric protein, meaning it contains a single polypeptide chain. And here we see two different representations of that. We have the ribbon structure on the right, and then some structural sequence showing here on the left. The heme group coordinates an iron. This group, or the heme, is buried in a hydrophobic pocket that protects it from being oxidized. It is in the Fe2+ state, and this charge on this iron will bind oxygen, whereas if this was oxidized to an Fe3+, it would not bind oxygen. So it is protected in this hydrophobic pocket. And here you can see this representation. This is the heme group. It's planar, and again, protected in this hydrophobic pocket. Myoglobin facilitates O2 diffusion in muscle tissue under high exertion. So when there is a high demand for oxygen in your muscles, say you're exercising, this starts carrying oxygen to those O2 depleted regions. It also, in aquatic animals, acts as an O2 storage molecule. You and I can hold our breaths for 1 to 2 minutes. Maybe some people can go a little more with conditioning. However, if you take a look at deep sea diving mammals like whales or seals, they can stay under a long time. And that's because their muscles contain about 10-fold higher concentration of myoglobin in their tissues than we do. So let's move on to another representation of this binding pocket. Now what we're going to look at here also describes the coordination of the heme group in hemoglobin. So it is the same coordination and binding pocket in both proteins. So here's our planar heme group with that iron coordinated. This forms an octahedron, or has octahedral geometry, so to speak. So we have nitrogens in the plane at 4 different corners of this heme, and it is when O2 is in the binding pocket, it is coordinated by the histidine in alpha helix E in the 7th position. And the iron group is also on the underside of the plane, coordinated by a histidine in the F helix in the 8th position. So there are 4 ligands within the plane, 1 ligand above, and 1 ligand below that coordinates and holds that heme group in place, or in the binding pocket. So what we're looking at here is an overlay of myoglobin and hemoglobin. So myoglobin, again, is monomeric, 1 polypeptide chain. We've seen hemoglobin before when we were talking about protein characteristics, primaries, secondary, tertiary, and quaternary structure. So hemoglobin, a globular protein, just like myoglobin, only it has 4 polypeptide chains. So where myoglobin is monomeric, hemoglobin is tetrameric. It has 2 alpha chains, which here are shown in blue, 2 beta chains, which are shown in red. And as you can see with this overlay of hemoglobin and myoglobin, it shows that their structure is almost identical. Therefore, they have evolved through a common ancestor through some kind of genetic mutation. Remember, we discussed the fact that structure does not always equal function. Sometimes it does, sometimes it doesn't. It's just not something we can assume. So it's kind of surprising when we compare the amino acid sequences of myoglobin and hemoglobin with respect to their alpha and beta chains in hemoglobin compared to the single polypeptide chain of myoglobin, the amount of sequence variability. In fact, the three polypeptide chains that we're looking at here are only 18% identical. So that lack of sequence similarity really highlights that important principle of protein folding or 3D structure. So certain tertiary structures, for example, that make up the backbone folding pattern of a globin polypeptide can accommodate a number or a different, quite a variety of amino acid sequences. But again, remember, structure does not necessarily equal function. Now, so if we take a good look at this, the purple highlights those residues. Remember, we call a single amino acid in a polypeptide a residue. Those residues that are fully conserved or identical. So we can assume from this that there's something very special about those residues that contribute to the function of these polypeptides and proteins. So there are interactions between the subunits in hemoglobin. There are two different states. On the left-hand side, we're looking at deoxyhemoglobin, so the heme group does not have oxygen bound. This is also called the T state. On the right, and this would be venous blood, on the right, with our hemoglobin oxygenated, we would be considered in the R state, and this would be arterial blood. So there is a change in the conformation when hemoglobin is in the deoxy state or the oxygenated state. And if you compare these two regions, look at these threonines. This histidine in the deoxy state is nestled between threonine and proline. However, in the oxygenated state, histidine has risen and is now coupled between these two threonines. So when oxygen is bound by hemoglobin, it alters the conformational state of that protein, as we can see here. That is called it is cooperative, but hemoglobin is an allosteric protein, meaning its shape changes upon binding to oxygen. It also alters the affinity. So remember, in hemoglobin, we have 4 heme groups in this protein. It also affects or alters the binding affinities of the other sites, and that makes this also cooperative binding. And just as a little aside, 1 ml of human blood contains about 5 billion red blood cells, each of which is packed with about 300 million hemoglobin molecules. So consequently, blood can carry far more oxygen than a comparable amount of water, for example. Like myoglobin, hemoglobin binds O2 reversibly. However, hemoglobin's affinity for O2 is lower than that of myoglobin, and we'll see that in a graph momentarily. I said the binding affinity for oxygen increases upon the binding of the first oxygen molecule. So after the first oxygen binds, the affinities for the subsequent bindings of the other 3 oxygens increases stepwise. So by the time that hemoglobin binds the fourth oxygen, its affinity for oxygen has increased 100-fold greater than it was when that first O2 molecule bound. Here we're looking at the oxygen binding curve of myoglobin and hemoglobin. The y-axis represents the fractional saturation. So that's the proportion of total myoglobin or hemoglobin molecules that have O2 bound. And on the x-axis, we have the concentration or the partial pressure of O2 in torr. Torr is the same thing as millimeters of mercury, if you remember. So this is the binding curve for hemoglobin, and you can tell that its shape is sigmoidal. And that sigmoidal shape tells me that we have cooperative binding. Comparing that to the myoglobin, which is a hyperbolic curve, we have this shape here. So we have hyperbolic for myoglobin, sigmoidal for hemoglobin. When the curve flattens out here, we are fully or as close to fully saturated as we can possibly get. What can we glean from this? In order to compare myoglobin and hemoglobin binding, we have to plot a hyperbolic curve for hemoglobin so we can compare these. So what we use to describe binding is what we call a P50, and that's where the protein is 50% saturated, and what we're going to look at is the pressure that's associated with that. So 50% saturation is right here at 0.5. If we go to the myoglobin curve, go down to our x-axis, although you can't see it because it's not represented in smaller increments, the P50 for myoglobin is 2.6 torr. Compare that to the P50 of hemoglobin, which is 26 torr. The pressure to fully or partially saturate hemoglobin is 10 times greater than that of myoglobin. So that also reflects the difference in the affinity for oxygen that these two proteins have. So how does this work in vivo or in our bodies? So myoglobin and hemoglobin both transport oxygen, and hemoglobin also transports CO2. So hemoglobin picks up O2 from our lungs, transports it to the tissue, hands it off to myoglobin, which myoglobin facilitates its delivery to respiring muscle tissue, and then hemoglobin picks up CO2 and takes it back to the lungs. So let's think of this in terms of oxygen concentration. So we know that hemoglobin has a low affinity for oxygen to begin with, and it requires a high concentration of oxygen in order to bind that first O2 molecule. Once that molecule is bound, the protein undergoes a conformational change, and again the affinity in the other binding sites increases, binds O2 molecules stepwise until it's saturated, and it can be then the O2 can be carried to the tissues. So in the lungs, the PO2 is 100 torr. Now, once hemoglobin has bound that O2, even though that binding is reversible, it doesn't want to give that O2 up unless we're in a low O2 region. So when hemoglobin gets to the tissue, the PO2 here is 20 torr, and that facilitates the release of O2 to myoglobin, who then transports that O2 to the respiring muscle. pH also plays a role in the reduction of or release of O2, and that is driven by the dissolved CO2, which forms the bicarbonate ion and promotes the stability of the deoxy state of hemoglobin after it releases that O2 and returns to the lungs. So there are a number, well, here are four particular amino acids that have been found to be critical for normal function of hemoglobin. Notice that what all of these amino acids do is stabilize the deoxy form, and there's one in each of the alpha chains, different positions, and one in each of the beta chains, also at different positions. You're not responsible for what the roles are here, but please note that there are these four that are absolutely critical for function. Now, there are about 950 hemoglobin variants in humans. Greater than 90% of those, which are usually single amino acid substitutions. So a substitution is going to be an amino acid that's replaced by another amino acid. 90% of those, or in most cases, those mutations are benign or do not affect function. So that tells me that the substitution is a conservative one that is an amino acid that is similar to the one that's being replaced. So it doesn't alter function. However, in other cases, there are mutations that result in very serious complications. I'm sure you're all familiar with sickle cell anemia. That is one. However, other mutations result in the decreased ability of globins to bind heme. If it can't bind heme, it can't bind oxygen. Decreased cooperativity. The protein could be locked in the T state or the R state. Increased propensity for oxidation of Fe2 to Fe3+. Remember, Fe2 binds oxygen, Fe3+ does not, and others could result in protein instability or decrease in protein stability. I mentioned sickle cell anemia. You can see that these are normal red blood cells here. They're disc-shaped and they have a little depression, and that sickle cell is this elongated and becomes rather fibrous. Here are some of those hemoglobin variants. The names are usually named after where they were found or discovered. Here we have Bristol, Hammersmith, Boston, Milwaukee, Kansas, for example. Again, you're not responsible for these mutations. It's just giving you an idea of what types of issues can arise from mutations. So here we have phenylalanine that is mutated to a serine, valine to aspartic acid, leucine to proline. Very different. So these are not conservative substitutions. They're very different changes in amino acids, and obviously alter the function of these hemoglobin molecules. One other thing I want to talk about that isn't on a slide, but that is the affinity that hemoglobin has to carbon monoxide. So we live in Minnesota, and at least one time during the winter season, I hear on the news of someone who has died or families who have died from carbon monoxide poisoning. So those detectors are really important. However, carbon monoxide, we all know it's a toxic gas. It's colorless, odorless, tasteless, so it's very difficult to detect. If you're only experiencing a mild or a small amount of exposure to carbon monoxide, you might feel lightheaded, a little confused, headache, maybe a little sick, flu-like symptoms. Larger exposures or significant exposure is going to lead to toxicity of the central nervous system, which can lead to death, essentially. Carbon monoxide is a competitive inhibitor of oxygen. Hemoglobin's affinity to CO is 200 times greater than its affinity to oxygen. So once it binds, it's really hard to get rid of it. You may know treatment in a hyperbaric oxygen therapy or treatment in hyperbaric chambers is the treatment for CO poisoning. What that does, it elevates or saturates the O2 in the body, in the tissues, to over 1,000% of its normal levels, and through applying pressure. And that is the only way that they can compete that CO off that hemoglobin molecule. There are other competitive ligands of O2. You're not responsible for those. I would certainly understand carbon monoxide poisoning, though, because it is a practical, unfortunate thing that happens in Minnesota and other areas as well. Other competitors are cyanide, sulfur monoxide, nitrogen dioxide. And this concludes the material that is on your first exam, which is, I believe, at the end of this week.